Hi, listeners. It's hard to believe, but New Frontiers in Functional Medicine has been going strong since 2015. With over 200 episodes and over 2 million listens, we are growing every year. My goal is to continue to bring you the best minds in functional medicine, epigenetics, and longevity. I want the content to be actionable for the functional medicine clinician and interesting for the rest of us. Thank you for supporting this podcast with your likes, with your honest reviews, with sharing the episodes with your friends and family. And if you haven't done so, please consider leaving a like or leaving us an honest review wherever you've accessed this podcast. Thank you, thank you, thank you to our sponsors. Because of our sponsors, these are companies that I trust, that we vet carefully, that we use in clinical practice, that I use personally. Because of them, everything associated with this podcast is free. If you haven't done so, I suggest you head over to DrKF to the podcast page where you can access the full transcript for every single one of these 200 episodes. And along with that transcript, you'll get an episode summary. You can get the research citations and additional content that is pertinent to the episode. I myself return to the transcript page all the time to pull out a little snippet of information that I want to recall or access a citation. Or better yet, sign up for our newsletter, again, at drkfitzgerald.com, and every single podcast episode will drop in your email as soon as it's released. Thanks again. Thank you to our Diamond Level sponsors, Biotics Research, Rupa Health, and Integrative Therapeutics. For over 40 years, the foundation of Biotics Research has been innovation and quality. Their goals remain unchanged: innovative ideas, carefully researched concepts, and product development with advanced analytical and manufacturing techniques. Biotics nutritional products are of superior quality and effectiveness. They're available exclusively to healthcare providers. Find them at bioticsresearch.com. I also want to give a shout out to our friends over at Rupa Health. They make lab testing easy, fabulous, doable for you, the clinician, and for you, the patient. Consider using Rupa as your super, super, super smart solution to all things laboratory testing. Visit them at rupahealth.com. For over 35 years, patients and clinicians have trusted Integrative Therapeutics for their dedication to quality, potency, and accuracy, and for bringing us exclusive products backed by science. From sourcing of premium raw materials to their meticulous manufacturing processes, they prioritize consistency, efficacy, and safety because our patients deserve nothing less. Find them at integrativepro.com. That's integrativepro.com. Thanks to our gold sponsors, Timeline, Mira, and OneSkin. Timeline, fueling cellular energy for a lifetime of healthy aging. Find them at timelinenutrition.com. Mira, the most trusted hormone monitor. Find them at miracare.com. And OneSkin, skin age reversal technology. Find them at oneskin.co. Hi, everybody. Welcome to a New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And of course, today is no exception. I am really thrilled to be spending some time with Dr. Nathan Price. Let me tell you all about him, and we will jump right into this really cool podcast. Nathan is Chief Scientific Officer of Thorne HealthTech and author of The Age of Scientific Wellness. Previously, he was CEO of Longevity, an AI intelligence company that merged with Thorne. In 2019, he was named as one of the 10 emerging leaders in health and medicine by the National Academy of Medicine. And in 2021, he was appointed to the Board on Life Sciences of the National Academies of Sciences, Engineering, and Medicine. He has spent much of his career, or his early career, as professor and associate director of the Institute for Systems Biology, co-director with biotechnology pioneer Lee Hood of the Hood Price Lab for Systems Biomedicine, and is affiliate faculty at the University of Washington in bioengineering and computer science and engineering.
He's done a ton of stuff, basically. He co-founded Araval after the Institute of Systems Biology, and he received a Healthy Longevity Catalyst Award from the National Academy of Medicine in 2020. He's got a ton of peer-reviewed scientific publications. You can get some of them in free full text, and I recommend that you do. In fact, we'll link some of them in our show notes. You've been influential in my thinking, Nathan. He was also chair of the NIH study section on modeling and analysis of biological systems, and is a fellow of the American Institute for Biological and Medical Engineering. That's a mouthful. Dr. Price, welcome to New Frontiers.
So much. You're so great.
So I've known you for a while. I've seen you speak many times, and you and I were just reflecting a little bit on your work at the Institute for Systems Biology, and I'm in functional medicine. I've been on faculty at functional medicine at the Institute for Functional Medicine for a long time. And, we sort of pride ourselves in being the clinical application of systems medicine or systems biology. Like, we like to think through that big lens. And I'm so glad that you've embraced us and that you've embraced functional medicine. I remember stumbling upon the Institute of Systems Biology many years ago and just being a little bit intimidated and overwhelmed, but also really excited that this systems thinking was in fact happening. And so I wanted to hear a little bit about that and about how you came to that, but also too, you obviously discovered functional medicine, and you must have been like, wow, there's actually people trying to apply this, clinically.
Yeah, it's such a great example, Cara, because it's like, I came from the standpoint of like a hardcore scientist, right? So we're building computer models and we're diving in, and it's all, NIH grants and just building that out. And as I got more and more interested in applications, and we became aware of functional medicine, I was in Seattle at the time, so IFM is there, and we became friends with Jeff Bland, and I could go on from and
all of us,
and then Jack and you and Laurie Hoffman, and anyway, on and on and on. And so with so many people, and it was interesting because we were very intrigued by this notion that, there's a bunch of doctors that believe, as we were coming to really believe, in prevention over treatment, obviously systems approaches where you're taking complexity, you're trying to personalize that to the individual. And it was interesting too, because we got pushback on some of this too, as you probably won't be surprised by, because you're very traditional doctors, they would kind of look askance at this stuff, right? They're like, well, they don't have the right clinical trials, and they don't have the, whatever. And this is somewhat anecdotal, but I'll just share it, because I started going to functional medicine meetings, and I'd go to traditional doctor meetings, and they'd be like, the science is all here and it's not there. And I would say things like, you know what though? When I go to a functional medicine meeting, everyone looks amazing. Everyone looks super happy. I got pulled out in one of these meetings at IFM, like onto a dance floor, which I rarely do. I'm dancing around like with, Mark Hyman is there. And I'm like, that dude has glowing skin. Like, he looks really healthy for his age, and all the other people that were there. And so I've said that on a number of occasions. I'm like, well, it looks like something right is going on over there, because you see that it's working. And as I've gotten more and more into this space, I've become very, very interested in how much personalization really matters. And I'm a huge proponent of randomized clinical trials and scientific evidence and all this. We can get into that. But I think there are ways to really merge personalization in really deep ways, especially as we get into our AI conversations. That is the kind of thing that lets us get really deep personalized evidence, but also taking this fundamental approach, because we know that one solution almost never works. But if you think about things deeply and you take a systems approach, you usually can have a benefit. And so it was just really fun to get into this world, and I've made so many friends in it, and it's been just fantastic for me.
That's awesome. Well, I'm so glad you drank the extremely healthy Kool-Aid, and that you joined our. And I'm so glad that you
Kool-Aid at functional medicine.
It's interesting. Of course, functional medicine has a long history of being challenged, but systems medicine, I mean, just the movement towards systems biology, thinking of being as whole and being an environment as whole. I mean, you guys had to have gotten some pushback also, like this movement to this expanded thinking is disruptive. And so, of course, if you go to a more traditional model where, there's kind of single variable interventions and, one molecular pathway, et cetera, it's going to shake things up.
There's no doubt. And in fact, pushback is one of the signs that you're on the right track. It's not the only sign. You can get pushback on stupid ideas too. So
yeah.
You need more than that. It's not the only sign, but it is usually a sign that you're doing something new. I remember when we first did, and we're going to get into this Pioneer 100 study that we did, and there was a big write-up about it in Science, and the headline, I forget exactly what it was, but the headline was something like controversial or, something like this. And I in my immediate response, I'm like, why not pioneering or spectacular or first of its kind? And I was speaking actually at a pharma company when it came out. And it was interesting because one of the executives there said something to me like, he's like, that's what you want. He said, if it's all positive, it means everyone agrees with you and it's not anything important. And it really kind of made me think about it. That actually, he's like, no, that means you did something that was important because he's like, that's how you know. And so I really, I appreciated that comment from him, and it was interesting because there's so many people, even in the farm, and we tend to, there tends to be a schism between, people. There's a lot of people in that world who really buy into the kind of stuff we're doing and love it. That machine isn't set up necessarily for it all the time because it does its own thing, but tons of people in there really love this kind of stuff too.
Well, and there's some pretty cool science coming out looking at sort of food-based compounds. I know like there's urolithin A is a molecule that I'm interested in, and kind of applying that pharma lens on it. So we can use the power, like since minds are beginning to expand, we can kind of use the power of their resources and their thinking and investigate, molecules that we know in our space are important.
Yeah, there's no doubt about that. And in terms of like Thorne, where I'm at now, like that's kind of our mantra, which is we want to bring the same level of rigor and science of the pharma and biotech space, but to the preventive space. And I think there's a lot of us that think that way because on its face, the notion that you can pull valuable molecules, interventions, and so forth from the natural world is sort of obvious, right? It's like, how could that not be true? And so, but you want to do that in a deep way, right? And that's what really systems biology, what functional medicine aims at and so forth, is that where we can take so many of those. And I really think about that as it's really like your first line pharmacy, right? A natural pharmacy. Because if you're having a problem, right, there's this notion that, well, what first can I look at in my lifestyle that makes a difference? On top of that level, well, what is there in the natural world that might make a difference? And then if that doesn't work, then, you get yourself maybe more into, pharmaceutical or that kind of intervention strategies.
I remember when you were recruiting for the Pioneer 100 study, we're going to talk about this. You did this at the Institute for Systems Biology, and I've tried gallantly to get on to be one of those 100. And I don't know, I was after it filled up pretty darn quickly. And I know
just a few extra people. It was eventually 108. We should
I should have begged.
We were just meeting back then. We would have loved to have had you.
It was so exciting. You were so exciting to me because we've been thinking through this systems lens, like we're doing our darndest to think through it, and to see you actually begin to bring that to fruition was just really exciting for those of us who've been trying to do this in clinical practice. But you're using AI tools, and we're going to talk about that more. You're sort of using where we all want to be. You're kind of vetting that technology and figuring it out for us. You actually, and you can talk about this, you were using a lot of the labs that that we use in clinical practice now, but you were just using this kind of bigger lens to investigate, and it was terribly exciting. So tell me about the Pioneer 100 project and what you guys learned, and we'll go on from there and what you've done since.
Yeah, so the Pioneer 100 project is something that Lee Hood and I initiated about 10 years ago. And the idea was that as we think about scientific wellness, which is, in our language, which is the notion that we want to bring the same level of depth to the understanding of wellness states that we have to disease historically. And one of the big aspects of that is you've got to generate all this longitudinal data on people who are by and large healthy. And, I'm sure we'll get into all the kind of things we've learned off that. I will say there was a ton of pushback against this idea, like to a degree that I found totally irrational.
Wow.
But people really didn't like it. And one of the big criticisms was that this was a giant waste of time, because they said, look, if you don't have an arm that is full of disease, right, to do the case control analysis, it's totally worthless. And that was a pretty general view. I was surprised by how vehemently people seem to hold that, because I'm like, I'm wasting my life, I'm not wasting your life. Like, what, if it fails, it's on me.
Yeah,
so
what's the threat?
What's the threat?
Right.
But so the Pioneer 100, but what we really had to get into then is, right, let's just get a general population. So we got, 100 or so people, mostly in the Seattle area. There were a few smatterings of people elsewhere, and took them through a journey where we said, all right, let's go through, and we measure a lot of the different labs that you would measure. I think it was about 100 to 150 of them. And then we measured proteomics out of the blood, metabolomics out of the blood. We did microbiome sequencing out of stool, and we were doing that on a every 3-month cadence. We also had wearable devices that people had, and then we had health coaches that would walk them through, like, how do you make improvements in your life? And we kept track of all of those kinds of things. Now, what came out of that was that we were able to show that people were able to improve their health markedly by going through such a program. And at the same time, we started generating this massive data. And we were really thoughtful about how we did the collections, how we standardized collections in time and space, which we could get into. It's a little wonky if we go through that, but there was a huge system that went through to enable that. And what it meant, and I think why so many functional medicine doctors and people in that space got so excited about it, is that it started to build this layer of information that we could put underneath really personalized interventions and start tying them to really deep science and tons of molecular measurements that at the time, no one had ever measured this many things on a population of people. And so from that standpoint, we called everyone in the Pioneer 100 pioneers, because they were the first people who were going through this kind of approach. And the vast majority of individuals allowed us to use those data for scientific discovery. And we've continued to publish, paper after paper after paper on them. We still are to this day. And so it's that was, I think, an incredible gift to science to be able to enable that to happen.
And controlling for time and space, just like the timing of the collection and the location, like just taking into consideration that, diurnal variation or like weather change, like were you considering all of that, location?
All of those kind of things.
Wow, that's amazing. That's amazing, actually.
Yeah, and so some of the ways you have to deal with it. So people, and I had spent a lot of time in my lab earlier, particularly with transcriptomics, studying these. We have a ton of papers on it, because you can often predict the lab that makes a set of measurements better than the condition.
Wow.
And I used to give this little talk where we'd have this cloud of points and we'd separate that and we'd see, like, okay, clearly this is cancer and non-cancer. And then I would say, I just lied to you. This is lab 1 and lab 2.
That's Joe's lab over there.
Normal and cancer in lab 1, and this is normal and cancer in lab 2. And you don't, and then you do the second mode in a principal component analysis, decomposition, and then it would start separating out. So there's all of these effects, and we don't know what causes all of them. There's just many factors, and you listed a number. Where are you at? What's the humidity? What's the environment in the room? Are there little differences in how somebody there's a million things. So what we actually did was to build a system such that when all of the vendors and all the different places where we would do measurements, we would spike in controls into every single one of those. So we would normalize them against something that we knew. So we actually had these big mixed vats of blood, and we would split them, and we would store them at each of the different locations where measurements were done, so you could spike those in. And then there were tons of algorithms on the back end that would control, so that your proteomics that you did in this is getting a little bit further downstream when we had the company, but you were doing on the, in South Carolina at 2 p.m. versus the ones that you ran in Southern California at, from a group in Southern California spread around, in the morning or the evening or whatever it was. And then all of that was tracked. And in fact, one of the big blood measurement companies had a spot on the board of Airvale, this, subsequent company, mostly just for that, so that they could see all of the things that we had done with their data and these other data types in terms of standardizing it and seeing how consistent are these place to place, location to location, time to time, and so forth.
That's fascinating. When we did our study out of the National College of Natural Medicine, I consulted Moshe Sef was an author, and just as a consultant for me, a mentor, as a epigeneticist extraordinaire. And he said that same thing to us. He said, you have to run the study. You can't do it remotely. You just really need to run it in one location, because DNA methylation is so sensitive. You need all your whole population just like being right there. He advised on that, and thank God we were able to do it. And then the other really crazy, because I'm not a scientist, I'm not in your world. Well, let me just say this one thing, and then I want to hear what you have to say. We had our specimen, our saliva specimen, processed at Yale, and when they put the specimen on the chip, I had to sign off on it, which meant that I had to go back to Moshe Sef and be like, what the hell? Like each position really mattered. It could make a difference just on that chip. So it was extraordinary to me how much needs to be controlled. And we were just paying attention to a handful of things, but I can appreciate all the variables you guys worked on. Yeah,
it's really true. And I think, the average person just doesn't appreciate how much variability there are in these kind of measurements. So I do want to make one point. So if you're trying to learn something in science and you want to get rid of all that technical variation, then you're right. You kind of want to locate it to one site. But if you're trying to do something like a diagnostic, if you do that in one site, what it means is that you could actually learn something that works in that site beautifully. But the second you take it anywhere else, it doesn't work. So I was on this committee workshop for the National Academies of Medicine, this like 10 years ago, a little bit more, that actually went through this robustness. And one of the things that we ended up putting into those guidelines were that when you did biomarkers, because there's 100,000 claimed biomarkers in papers every year, 2 on average make it into the clinic. So that conversion rate.
Wow.
That used to be a little bit of a point of pride for Lee and I, because in one decade we had 2 of them, so we felt pretty good about that.
That's awesome.
So anyway, that was following that report. But basically, it's so one of the things that you want to do is to deal with that uncertainty up front, so that you're actually gathering data from multiple sites and you're machine learning across that, because that enables you to get signals that are more robust for that transplant. So it really depends kind of what your goal is. I don't get too wonky on that, but that's
well, it's great. It's very compelling. Yeah. And of course, I mean, you want CRP in Miami to be, as good as useful as a CRP in Connecticut. No, I completely get it. We were working, ours was a pilot investigation, and we just, we had to.
Totally. It is, if it's a small study, and
yeah. It's a different conversation.
And you have the cases and the controls from different places. And that happens all the time for practical reasons, and it's really problematic from a
very disruptive. Yeah, of course. I mean, I can just see in ours, when we compare the cases and controls, we see different, and and these guys were like recruited from the same locations. Anyway, I just want to say for this clinicians, functional medicine clinicians are the large portion of the audience here. And so you guys in the Pioneer 100, you were using labs that I want to just say we use all the time, standard chemistries from, I think, both LabCorp and Quest, but, Genova tests, stuff that we're ordering. So I want to just underscore it, because, you've since harvested a ton of information, and we're going to talk about some of that research. But it's validating for us, I think, that we're, in the right direction. It's nice to hear, or it's nice to, go ahead.
And those standard clinical labs were so critical, because when you start measuring the microbiome, you don't know that much about the health effects. And you start measuring the metabolome, and you only kind of know health effects. And so it's like a Rosetta Stone in many ways, because you go in, we'll get into this when we get into specific, areas that we want to talk about. But the you start to learn, well, how does the microbiome affect metabolic health? Well, you've got to have validated markers of metabolic health that you believe in order to ask that question and so forth. So it really is a way for you to interpret new data types, is to have these other ones that you really understand much better.
But then you moved into looking at thousands of individuals. So you were applying this technology. I mean, you guys were for sure, ahead of your time. You were really early in this world, and we were excited about it. Those of us kind of early adopters in functional medicine. But I mean, I have to believe that is a big reason that you didn't accelerate, that Arivell wasn't able to, isn't here still today. However, you had a massive data set of almost 4,000 people, and the Pioneer 5,000, amazing. And you learned a ton of stuff. I mean, I quote you in my book. I mean, I've just paid attention to your papers. They're incredibly valuable. I just pulled open actually for this conversation your 2019 publication, Multiomics Biological Age Estimation and its Correlation with Wellness and Disease Phenotypes, a longitudinal study of, at this time, 3,500 individuals. And you published that in the Journals of Gerontology. This is a awesome paper, you guys. This is awesome. I'm pretty sure it's free full text. I'm not, well, anyway, there's a ton, there's a ton of information in this. It's so yellow highlighted. So you guys learned a lot from this about biological age and chronological age, whether biological age is even manipulatable, if we can slow it down, if the association with biological age and disease. You also began to really harvest the biomarkers around this. And to your point, standard chemistries that we're looking at are featured broadly in here, and really they're kind of linchpins in looking at bioage. So I want you to talk about all of that, and then you put numbers behind, the accelerated aging in certain disease states. Like, I mean, I've quoted you many times, over 6 years on average, the diabetic patients' bioage is ahead. So it's just a great paper. I recommend anybody to get it. It's influenced my thinking quite a bit. So just talk to me about it.
Yeah, so it was an incredible study. John Earls, who was my graduate student at the time, was the first author on that, and did a ton of work on it. So there's a number of take homes that were, I think, very fascinating. So first was, how does this biological age measure change over time in people who are aiming to improve their health? And I think it's really important to keep in mind that we develop the biological age later. So we're not targeting to it with the coaching in Arivell, but we develop it, and then we go back and look at that. And it turned out that on average in that population, people got better in terms of their biological age compared to their chronologic age by 1.16 years per year on average.
Amazing.
They're getting.
So is that escape velocity? Are they like going to live forever now?
No. So if it were just a pure linear trajectory, yes. Sometimes you hear people talk like that, but the larger that delta gets, the harder it is to maintain, right? And there's limits to this.
Right.
And
so basically the healthier you get, it's slowing down considerably. That's what you're saying about the delta.
I mean, sometimes at parties you'll joke about that. I'm going to go out the backside. I got to get worried because I'm just going down like too fast or whatever. But that's not a real worry. You're not going to regress to childhood, at least not yet. We don't have any technologies that way. So but it is a pretty good measure of health. But there's a window of what's possible today. You can't do it forever. Certainly at this point.
How broad is the window? Have you figured that out yet?
Probably play with about 20 years or so.
That's a good chunk of time.
Which is a good chunk of time.
Mm-hmm.
So just, so it's 1.16 years on average. Women did particularly well. So women in Arivell were getting better at a rate of 1.5 years per year. So they were getting, half a year younger on average on this metric. And men were getting better at 0.8 years per year, meaning that every year they age on average 0.2 years, according to this metric. And so we saw that. For myself, I'll even say, so I started with a biological age that was right around my chronologic age. And I was in the program for 4 years, and my biological age dropped by 10 years, according to this metric.
Incredible.
Which was which I have to say, just to share a personal, that was very gratifying to me because I had kind of felt like my road through Arabelle, I felt like it had helped me in many ways. And cognitively, I was feeling better. I had changed lots of habits. But one of the things that I had really wanted to change more than it did was my weight. And my weight had been pretty stubborn. Lose gains really rapidly, lose gain, So I was doing kind of the usual thing. But what was fascinating to me is, at least in my blood chemistry, it just kept ticking younger. And so when that came out, because I didn't know who I was when we published the study, I went back and I got, because I'm in there, but I have a random ID just like everyone else. So I didn't know who I was, but I was able to get it. And I had John look it up and I said, send it to me. How did it come out? And I was so pleasantly surprised. It was one of the reasons we ended up taking the clinical lab version of the biological age out, similar to what's in that paper, and turn it into a commercial product at Thorne, because I just thought that was, it made a big impact on me just to be able to see it. And so that was was something that I think was very exciting from the standpoint of change. Now, another thing that we get into in that paper that is, probably not appreciated as much, and it is an important point, is that you can build biological ages from various different sources. Epigenetics is the most famous, but you can do it from metabolomics, you can do it from proteomics, you can do it from clinical labs. The clinical lab-based biological age today is the most related to clinical outcomes. And it's also important to note that these ages are only somewhat correlated with each other. So if you correlate them straight up, they're about 0.75 correlated, well, maybe even 0.8 correlated, but that's because they're all strongly correlated with age. If you only correlate delta age, right, the difference between biologic and chronologic age, they're only about 0.25 correlated, which means that they share information, but there's quite different information you're getting off of an epigenetic age versus a clinical lab age versus a metabolic age versus a proteomic age and things of this nature. And we're just starting to sort through a lot of that.
Well, tell me why using clinical biomarker-based bioages are more associated with clinical phenotype. Like, how did you establish that?
So I would say that we looked at that certainly in the context of the metabolomic, proteomic, and epigenetic. I'm pulling partly that, though, out of the work, a paper that was co-authored with by Morgan Levine and Steve Horvath, where they did a direct head-to-head comparison of epigenetic age versus, phenotypic age or age based on, clinical labs, and compared that to outcomes. I think it was in the NHANES study, and basically did an analysis of that, and then found that out. There are, of course, interesting arguments for for epigenetic ages, and some of the ways. And there's a lot of cool science that's happening on that. I haven't been terribly involved in that, but it's a huge aspect of the aging field and so forth. So that's what I'm saying. And so I think in some of these biological ages, it really comes down, and the first clinical study you did was actually a really good example of this, which is the need to make them actionable. I think is so important, because I feel like if you're talking about an omics where all it's going to tell you is your age back, it's kind of an interest. To me, it's kind of gee whiz, right? It's like, it's cool, interesting. It's not hugely meaningful. But when you can tie a biological age with insight into what's driving it, and a plan of action and a way to improve your health, then it's super, then it's a driving force for preventive medicine.
Yes.
Scientific wellness or healthcare or whatever,
Well, and what you guys did that's really super cool, that I want to kind of underline an exclamation point, is that you didn't consider biological age as part of your intervention. So you guys cast this wide, biomarker net. You did your interventions, which we should talk about. And then retrospectively, you kind of analyzed, you layered the clock onto it and saw these really cool findings. Because I think what's happening now in this space, there's just this obsession with biological age. And, there's just this, there's a lot, I think there's a kind of an attachment now to sort of treating the test, and not the human, or attempting to manipulate the test, with certain interventions without, considering whole person. And so, to your system's roots, you just turned that upside down. That's huge.
Something you have to be really careful about. I forget who said it, but there's an adage: whenever something becomes a target, it ceases to be a good measure. And so And what that means is that as soon as you're kind of trying to manipulate a variable, and so when we were doing the biological age and in the paper, one of the things we tried to be very careful about is don't include in the score any of the kind of things that people supplement with. Because obviously, if you're just trying to go to a score and it has molecules in there, right, you could supplement with those molecules directly and you could manipulate that score a lot.
Yeah.
But is that meaningful? And it might be health beneficial, but you haven't proven it by the same measure. So what you have to do is tie that to these other health measures, because what you actually care about is the degree to which the delta age, right? The age different. I don't know. I'm an engineer. Delta is how we talk, but if there's the age, the difference between, your biological and chronologic age, to what extent does that show you that you've improved or hurt your health? And then and then tying that to a plan of action, like what do you do about it? And there's so many axes to dive in on there: chronic inflammation, oxidative radical, you could name them all off, cognitive reserve, on and on and on. And so what I really love is this whole notion of having metrics for wellness. So not just, do I have cancer, do I not have cancer, but actually, and I think biological age is a great first example of this, but just measures that say, am I more metabolically flexible? Am I more metabolically resilient this year than I was last year? Is my cumulative inflammation burden lower this year than it was last year? Is the the metabolic deficit, or my excess capacity for energy generation in my brain, is it better this year than last year? Things that you can focus on to get better and better at, that make transition to disease less and less likely. I think those kind of things are super important.
Yeah, extremely important. And so the clock that you guys have at Thorne, I mean, there's very tangible action steps that one can do based on the findings.
Oh, yeah, absolutely. And one of the things that I'll bring up, with Aravale being early, so I remember someone commented to me and they said, "Oh, I wish Aravale was still around because I want to do that microbiome test." And my immediate retort was, well, Thorne's microbiome test is about 100 times better than what we had at Aravale, because it's
later. Early. Yeah, that's right.
And back then we had to do 16S sequencing.
Yeah.
And you could look at diversity, but it was fairly limited.
It's crude. Yeah.
Now you can get into a lot of detail. So, it's just so actionability, right? So off of a microbiome test, you can look at things like, metabolic pathways. So if you're looking at different gene complements, are you making too much ammonia that might make your stomach too basic and, cause you digestive issues? You can see those kind of things. We did a study, this was joint with colleagues at Sean Gibbons in particular at ISB, looking in that population at who went on to lose weight versus those who didn't. And when we looked at baseline, once you controlled for BMI, there was not a strong signal in the metabolome or the proteome or even all those clinical labs, but there was in the microbiome. And so the microbiome was quite different for people who went on to lose, in this case, I think we had it at 1% of their body weight per month, and those who didn't. And the big difference was, if you had a microbiome that broke down complex carbohydrates into simple sugars, those individuals did not go on, by and large, to lose weight. But if you had a microbiome that broke down complex carbohydrates preferentially into short chain fatty acids, was much easier. And there was a big difference between
Fascinating. Those. Wow.
There's many things and that one, I'd want to replicate in a prospective trial.
Yeah.
But there are mechanisms that you can look at that make quite a bit of sense, and that you can dive into and understand in that way. Anyway. So
and all of this will funnel ultimately towards lowering your biological age, reducing disease risk. That's fascinating. What were the organisms, out of curiosity, were you able to nail down, or was it just sort of action as a whole?
It's across a bunch of different organisms. So we weren't really looking at it from the standpoint of organism by organism. It's really
just function,
metabolic, the gene content.
Okay.
And in fact, in a different study that we published in Nature Metabolism in 21, there we were looking longitudinally at aging. And it turns out that if you stay healthy starting around age 50, your microbiome starts looking less and less like anyone else's microbiomes. So if you just compare them, they start being more and more distant from each other. And yet the metabolic transformations that are associated with healthy aging are largely conserved. And so they're solving these similar issues, but in very diverse ways. And so I think in many cases, we really want to get at like, what's the metabolic function of your microbiome as you measure it in the metagenome is much more predictive than our, the actual species. And there are some differences to that, right? If C. diff is obviously important because it secretes certain toxins and, there are areas that would you get into, but broadly, those metabolic components are more predictive.
So fascinating. Well, we know that, bacteria just share genes like, I don't know,
all the time,
playing cards or whatever. Like, so yeah, it's just really, really interesting to me. All right, what do I want to ask you? So interesting in this, going back to this 2019 paper, I included it in, I created a table in the book of some of the top labs that we can get, that people who are reading it can go and ask their doctor for. And I was definitely looking at what you were seeing, because you had some standard biomarkers that any of us can ask our physicians to order, or a panel that we can put together as providers for our patients, are really pretty strongly associated, beneficially or, inversely with biological age. Can you talk about some of those standard chemistries? What are your favorites, and has that changed since this publication?
Yeah. So the ones that really matter the most are not terribly surprising. So hemoglobin A1C, more so even than fasted glucose, but both of those, because metabolic health is number one driver. And I'm sure this is what you talk about with patients and so forth, because you've got to be metabolically healthy. That is a huge driver. Just to give a comparison, smoking, as I recall, raised biological age by 2 years.
That's it. Yes, I just read that. That was actually my next question. Like, what's up with that? I mean, really?
Less than we would have thought. But metabolic, so if you're smoking or you're pounding potato chips, I don't know, or sedentary, right? Being sedentary is a huge killer, as we know. So that metabolic health is like the primary driver. Second big driver is our hormones. Now, we don't get into a ton of detail in hormones in here because we didn't, measure tons of them, but we did it through the reserve for many of the hormones, which is DHEA.
That's
and
right.
DHEA turns out to be really good from a biological age standpoint, because especially if you look at, female hormones, because they cycle and change so much, it becomes, you can do it, but it's a squishy target in order of trying to create like a robust biological age score. And so DHEA is quite, and it's a pool for both estrogen and testosterone. And so that turned out to be a very big measure. And then a third bucket that really was important were toxins. So
yeah, talk about those. Which in particular?
Lead was the one that we really found a lot. And so one of the reasons for that is that you're not very well able to clear lead out of the body. And so it ticks up over time. And so one of the big factors of biological aging was just watching that increase. And of course, there's all kinds of toxicities that come along with that.
And let me just say, it's increasing, not necessarily because of current exposure, if that's what folks are thinking about. It can be liberated from bone, because that's where we sequester it to kind of prevent the toxic effect of it. So we can see this, what we call secondary half-life in circulation.
A great point. Yeah, exactly. So you could have even removed yourself, but it's continuing to tick up. Mercury was another one. And mercury can old style fillings can leach out. Actually, our CEO at Araville had one of those. Like, he found it as he got in Araville. He's talking about this publicly.
Wow.
So he actually got those switched. Tuna sushi was a big one. Jeff Bland, he shared it. Well, he was in the study. He was
he was open about
shared that publicly, so I could say it. And realized, okay, got to back that off. We had 3 people that were really high in the Pioneer 100 on mercury. 2 of them because of tuna sushi primarily, and the third for the tooth. So those become really big issues. And if you start tying this in with genetics, it can become even more important.
Interesting.
What we found, because one of the things that was really struck me as just kind of a eureka moment early on was
Yeah.
that we didn't actually have to just watch disease arise and go back and try to look for early warning signs, but we could use genetics as a rubric to look at things right away, because you could take data that we were getting already on people, and because we have genetic signatures that are associated with high to low risk for all kinds of different diseases, you could use the genetics themselves to just look in the population of healthy people and start to see what's different between people who are at high and low risk for all these things.
This episode is supported by Practice Better, a leading all-in-one practice management platform. Most health and wellness professionals love what they do, but feel overwhelmed with time-consuming administrative tasks. With Practice Better, you can automate tasks like onboarding, scheduling, charting, nutrition planning, and billing to help you better focus on the parts of your practice you love. New Frontiers listeners get 20% off the first 4 months of any paid plan at practicebetter.io with code KF20. Visit the podcast page on drkfitzgerald.com for more information.
So one that comes to mind is
Yeah.
ALS. And so people who are at really high risk for ALS, one of the things we found is that they tended to have more heavy metals in their blood. And so when we looked at that, that's interesting because heavy metals are a known risk factor for ALS, but we were finding this in people that don't have the disease, right? But just as a function of the genetics, which suggests that if you have a polygenic risk score that tells you you're at high risk for ALS, you're probably less well able to clear heavy metals than the average person. So you should be more diligent on that. And we found 750 examples like that. That's just the first.
Amazing. Did you happen to, so, one of the reasons we can accumulate lead or toxic metals is if we've got, one of the genes or we're heterozygous for anything associated with hemochromatosis. I don't know. Did you look at that perchance? Because that increases the quantity of transport proteins in the gastrointestinal tract, actually in the brain as well. And we just absorb easy because the toxic metals can hitch a ride on the iron transport proteins. Anyway, something I've observed clinically.
Absolutely. And it's a really great example. And hemochromatosis, because it is, the most common genetic disease in Caucasians, but it is, but basically, we had people in the Pioneer 100 like that.
Yeah.
The people that we screened had sky high ferritin levels in their blood. And so we got them in, they really credited us with making a huge impact on their health because he had become quite debilitated and didn't know why. And it's the kind of thing like, well, your doctor should have measured that and his doctor should have, but he had concierge medicine on both coasts and I don't know. They never looked at it. Who knows why?
Yeah.
But it was, but we found that. And the treatment for that, of course, is very simple.
Yeah.
Yeah. You go, it's the thing I like to say is we did this huge high-tech study to find the one disease you can cure by leeches, but it's, it's
a huge, it's a very underappreciated, anti-aging intervention, super potent, and it benefits the world tremendously if you give a little blood. I just got a CBC and, I donate blood and just noticed that, it's time. My numbers look good. Did you happen to notice if he also had metals, kind of track toxic metals tracking with that elevated ferritin? We'll have to go back and look.
He did, as I recall, he also had high, relatively high mercury and so forth.
Yeah. Those
the others I don't remember for sure right off the top of my head.
I published a case report on that topic from an IFM faculty. Actually, I wrote a whole book over a decade ago and collection of case studies, and that was one of the most interesting ones. Just seeing how those toxic metals can hitch a ride. It's so interesting. I also want to bring to the attention of folks what you guys found were the standard biochemistry, the standard biomarkers actually track really nicely with the PhenoAge DNA methylation test created by Morgan Levine. I think you were referencing that earlier, and Steve Horvath.
He does a lot of great work. Yeah.
So if anybody's familiar with the PhenoAge, the DNA methylation score is based on albumin, I think lymphocyte count, if I'm not mistaken, alkaline phosphatase, mean cell volume, just a handful of standard labs, CRP. And you found that, we
find similar things.
Yeah. Yeah, you really did. And so these are things that we can look. And then you actually added on that. I'm looking at the tables in the paper here, a lead, A1C, et cetera.
And it's worth
So-
noting that those were, learned and regressed independently. In other words, it's not like, so if your doctor's bringing that up with you, yeah, there's something to it. Like you can have independent labs that find, that will find that. And if anyone else dives in and does it, they're going to find it again too. It's a very robust signal.
I want to just bring your book to folks' attention. You cover a lot of this stuff in the book, and it's just really easy to read. It's incredibly interesting. You wrote it with Lee Hood, who, if anybody doesn't know, he was one of the guys who mapped out the human genome. Am I correct in that? Like he's
Absolutely.
He's a pretty extraordinary human as far as I know, sort of from a distance. But you guys,
yes. Absolutely. Yeah.
You guys write in chapter 10, the AI, the artificial intelligence imperative. You write, the realization of this vision of healthcare that is predictive, preventive, personalized, and participatory depends on many moving parts, but no tool will be more important than artificial intelligence. So you're clearly using it all over the place in your work. When is it going to be, in my clinic? Like, when is it going to be like real time influencing us? I mean, how can we access, AI?
Yeah, so I think it's worth saying that AI is already being used in many ways. And then maybe I'll talk about short term and longer term.
Yeah.
So one of the ways that we chronicle in the book that AI is being used right now is the correction of medical errors. So some of that comes around, as you're filling out an EHR and you're going to click on a drug, for example. These drugs have long, complicated names. And some of them that have very different indications sound pretty similar.
Mm-hmm.
So a busy or tired or, physician could easily, and do, in fact, at some level, click the wrong box, whatever. So one of the ways that AI is used, and this is a program called MetaWare, just simply corrects for those kind of things, which just brings up, oh, did you mean this? Because it understands that it looks like this patient needs, has kidney problems, and you just clicked on something that is, for a heart trouble or something.
Yeah.
And that has already been able to fix hundreds of thousands of other potential errors in the system. So there's some low-hanging fruit that are just useful for that level. Next level up, just looking at the kind of AIs that have burst on the scene lately with ChatGPT and other things like that. There was a survey, and I believe the numbers I saw were something like 65% of doctors say that they have used that as a tool for, as they're doing diagnostics and things of this nature. Now, it's not at a level where you would completely trust it and those kind of things,
Sure.
but it is very useful if you're trying to remember. And I use it myself, I have to say, when I'm like, oh, remind me about this or what, and if it's important, I'll double check on it.
Mm-hmm.
But especially the later versions are pretty good. Like, so the ability to do that, and coming very soon, I think, is going to be a big reduction on burden of physicians for chart notes and things like this, because you can set up a recording, it can listen to you talk, it can transcribe, and then you can have GPT write up notes. And then the doctor is now just reviewing the notes, right? Reviewing and editing, but hopefully that's a 1 to 2 minute thing instead of however long it takes. So those kind of things are important. Another area that I think is going to come in small pieces and then large pieces are digital twins. So this is something that's talked about a lot. And this is one of the things we've actually developed at Thorne with along with a partner around digital twins for brain health. And this has been, and this is what I've spent a lot of the last 3 years on. And it's been pretty remarkable because, one, it's given me a very different view of what drives Alzheimer's disease, not amyloid.
Right.
And I think it is really driven primarily by metabolism. And there's many ways that basically by the need of the brain to create energy. So your brain consumes 20% of your body's energy. It's 2% of biomass. So it's 10 times more metabolically active than an average tissue. So it's a massive energy hog. So you have to feed energy up to it all the time. And if you starve mice, for example, the last two areas that get, it will start muscle wasting. It pulls energy away from everything until the brain and the heart. And at the very, very end, it preserves the brain, number one. Right. Now the brain makes decisions, so maybe that's not surprising, but it is the energy hub.
Amazing.
And so when you start looking at this, like, what does it take for a neuron to stay, to stay alive? Then you have to satisfy that. And as you get older, your ability to perfuse oxygen into your brain goes down. And then and it turns out that under these conditions, you want to keep cholesterol levels and astrocytes low. ApoE has a role in that. ApoE4 will transport that cholesterol, but slowly, which keeps concentration high, which keeps energy production in neurons low, which means more neurons die, which means you get Alzheimer's at age 70. If you have ApoE2, it's fast, keeps that concentration low. You're more efficient at making energy efficiently under low oxygen, and you don't ever get Alzheimer's. So you get it when you're in 95 or something. And so these kind of changes, anyway, I don't take too much of an aside, but you can get into these things. And what the digital twins have let us do is we can then take this mechanism, and we've layered onto that core mechanism the effects of inflammation, the effects of microglia, the effects of TREM2 variants, the effect, vitamin D, phosphatidylcholine, every known risk factor that we can find into a unified model of one single complex mechanism represented by this, sophisticated ODE based model with the Bayesian network overlay. But basically with this complex model, and then we've been able to simulate 10 million digital twins of patients. And when you do that, you're able to recapitulate very closely the age of onset of all the different genotypes, match all the risk factors. So we have a pretty good representation. We think we're going to, we've compared it to 30 clinical trials and research studies that are out there. We're going to be doing a prospective trial on it. But basically what it does is it gives you this really holistic view. And these digital twins, and we're going to get that out hopefully to clinicians. I hope next year. We got to figure out the path forward and all the regulatory hoops we got to jump through and all those kind of things. But we'll get that kind of thing. Is going to be, it already functions, it works. We can use blood data. We can make these measures. We can show them to people. We've shown them to a number of physician groups. I'm going to give a talk to the National Institutes of Health, Alzheimer's disease and combinatorial therapy. I'm lucky enough to be one of the 3 opening plenary speakers for that meeting, and it's going to be all about this, and we're going to talk about it.
So so the digital twin model will be available to us in practice?
Yes, that is what we're going for.
Wow.
So that I think is going to be really important. And so as we do that, and so we're going to be able to do that for the brain. We're going to be able to do it for metabolism. We're going to be able to do it for skin health. We're going to do it for aging. Like, you can dive in, and it gives you a rubric to understand how molecular level data relates to patient outcome. And you can do this in really powerful ways.
So we would be able to, in advance, so we've uploaded all the patient data that we've got at our fingertips, and maybe you're going to guide us to order certain things, like we want a full genome and we want this and this. But then we can try different interventions for what's going on in this digital twin model to see how,
and you can simulate the likely outcome of the intervention in advance.
And the combination of interventions and interactions. So you could see like synergy of an appropriate combination or detriment. Or
yes, for the areas where we have enough, detailed science, and we've taken about data from about 1,000 papers to build this thing out. But as you take that information, then that is going to let you be able to ask the question, like, so what's the best intervention? How many years? So what this thing will do is actually, given the data, it will say, here's the most likely age of onset. And there's a probabilities associated with all these things. We try to take into account measurement error, error in, like, how much we know about, the effects. So there's a propagation, right? So it's a probabilistic model, but you get a range. So, we might say for this, there's an example I often show, but it's, a 50-year-old man, and we, in this case, predict age of onset would be 61, most likely, but there's like a 99% chance that he's going to have it before 68 if he doesn't do anything to change. But then you can give a set of interventions. It will then say, whether or not the patient is likely to, what percentile responder they are to each of them. Oh, and this is coming back to our earlier discussion on functional medicine. One of the things that stands out is that very often all the individual interventions are predicted to do nothing. Very often. But combinations are predicted to do a lot. Like, so you might take something where every individual intervention is predicted to do nothing, and yet there's a combination that will give them a decade of extra life. And we can simulate the quantitative mechanisms for why that is in every individual case for the areas, again, where we have, enough information. And so one of the other things, and I gave a talk to the advisory council to the NIH director last month on this same topic, basically arguing that we should do prevention trials not on single compounds, because when you do prevention trials on a single compound, almost everybody is a non-responder. And especially when you're talking about natural compounds, the N that you would need and the length of time in order to see that, because you're just and the economics make no sense, because you're not, if I do that for vitamin D, it's not, I'm not going to make, the company's not going to make $100 billion on that over time. So I cannot justify the length and type of trial it would take to do that analysis. But if I test instead the digital twin, which is a representation of everything that I think I know about the disease, where I'm going to take measurements, I'm going to run it through the digital twin algorithm, and I'm going to do something different, personalized for every person. Now, the reason that approach in the past has not taken off amongst like your hardcore scientists is because it comes off as too squishy too often. Because if you go in and you say, well, I'm just going to do the thing that I think is right, it's hard to say, well, are you changing it post facto, right after the fact, or what? But when it's written down as an algorithm, it's still one concrete thing. The algorithm doesn't change. It is being tested. But the fact pattern against the algorithm means that you can personalize the intervention to every single person. And when we've run this analysis, it shows that we should be able to get clinically validated improvement in things like mild cognitive impairment leading to Alzheimer's in a 1-year trial with maybe a couple hundred people, instead of a trial that on a single compound that would take forever. Because now you're talking about trials where most people are responders, instead of trials where almost everybody is a non-responder. And it's radically different. And so one of the things, I gave this talk at the American Nutrition Association meeting last month, because I won their science award for this year, the Alexander and Mildred Seelig Award. And so gave the opening talk there and got into this because for the nutrition field, as well as for functional medicine, to me, this is like the holy grail, because what we can do now is say, and this is where those old discussions on, like who has the scientific high ground, what we can actually say is, no, we can actually run randomized, rigorous clinical trials on individualized interventions. And the way that we're doing these in the traditional approach is not as good. In fact, it's not a very smart strategy, because if you're isolating that one compound, you can never get to the end that you want to get to. You can never show the effect. You have to do these multimodal trials. And so that's not a new, right? We know that. But by writing them down in the algorithm, you can really make that incredibly standardized and I think scale it. And so what I want to do is to say, let's build those for every single major category of health, run all the trials. And now all of a sudden we have personalized evidence-based medicine that's incredibly rigorous and actually at a scale of rigor that's better than what we have in the traditional approach. Still in RCT, but it's personalized. And so that I think is going to be, I don't know, I get very excited about this topic because I just think that is the way that we can have an incredible
mind-blowing. Wow. I was talking about Bob Roundtree. I was texting back and forth.
Bob.
I'm about to get on I'm about to podcast with Nathan. Any thoughts? And he said, ask him about Alzheimer's and the digital twin. And I'm so glad. I mean, this obviously could and should maybe in the future just comprise a whole conversation, because it's extraordinary. So if I'm understanding it, like if I just really simplify it, if we had it in clinical practice, so that there's the algorithm is consistent, but what changes are my patient's data, because obviously their data is going to, from patient to patient. So all of that, it could be, obviously tons of stuff, environment, age, blood biomarkers, et cetera. All of that is packed in there, but the algorithm is consistent. And then the interventions, obviously, just based on filtering through their individual phenotype, will shift the response to the interventions. Is that basically what the digital twin is?
Exactly. So, so you might come in and it will say, for you, you need vitamin D and phosphatidylcholine and, and to improve your sleep profile by this amount and whatever, and you'll get 8 extra years. Wow. And for me, it might say you need to up your VO2 max and take phosphatidylserine and, and lower your basic inflammation or whatever. And it will be some combination of things based on the biology that it observes.
How are you guys establishing dosing? Like, how do you know, and are you staying within the boundaries of, what's been studied, or are you jumping outside of that?
So we are staying within the boundaries, and that's something we honestly could do more of as we go forward in terms of the dosing. So we try to calibrate it to papers around, like, how much dose gets in, and then, what the effect, and so it might hit a receptor and then like change it. One of the nice things in the digital twin models, the style that we're using, is because it has this Bayesian network overlay. What that means for people that aren't familiar with Bayesian statistics, which is probably most doctors, is that it adapts itself to as much or as little data as you have. So in other words, if I know nothing about you, right, you're a human, it will output the average response for a human. And if I say, this is a woman, it will update to the average for a woman. I say 48 years old, it adapts to that. And then I start saying, who's pre-diabetic and has been pre-diabetic for this length of time and is ApoE4 double positive, and on and on. And it adjusts the parameters and then what the simulation does with however much or little data as you have. And so that's kind of the beauty of these kind of approaches, because we think we can use that in, with doctors, based on the amount of information they have, what they're doing with their patients in a relatively flexible way. It's not infinitely flexible, of course, because we have to have hooks into the model of ways that we can simulate that, right? So if you have like a new hypothesis that we haven't contemplated, right, the model won't magically be able to do something with that. But as those areas emerge, we can build out the biology and tack it on, and it can become smarter and smarter. And this is one of the things that I really want to build going forward, is a network of physicians who are using this and sharing data back. Because what we would be able to do is create a community where we're learning at an incredible rate. We're able to do periodic clinical trials around this, and update. But basically, it allows for a community learning system where we get better and better at the disease. And of course, what we're all hoping is that, we ultimately get to the place where we can really effectively treat all of these conditions. And what we want to do is build, the back end, the algorithms, the digital twins, provide tools and things like that, and then let the doctors build the programs and work with the patients the way that they want to. But that's what I'm really excited about, because I think we can get to this world where we get, dense measurements that can be done cheaply and easily. And, we spent a lot of time at Thorne on that too, like our microbiome wipe to make the, sample of microbiomes easier. And we've got this painless at-home blood measurement device, the nano, so we're trying to make these things like simple. But then what I really want to do is just to tie all of that information together with these deep biological models, where we get these digital twin simulations and then make them readily accessible to the community, and have a lot of back and forth dialogue in terms of what are we learning and what's working and what's not, and iterate. And I really believe that the pace of progress for chronic disease could be majorly accelerated by building something like that. So that's my
100%. It feels like logarithmic. When I think back on the beginning of my career, I've got like the Boehringer Mannerheim, wall chart over there, like just sort of trying to hold all of this in my head when I'm interpreting lab data and thinking about mechanisms. I mean, you've just blown it open. That's really, just really fascinating, really exciting. God. Yeah, I would love to, if you need a clinician, I would volunteer.
I will reach out to you.
Yeah, I would just love to be a part of it. We've got a ton that we could keep talking about, but I would like to have you back maybe, I don't know, when you're nearing launch or whatever, but just a couple more questions on this digital twin. Like, nutrition information, how granular can it get on macronutrients, et cetera, or combinations? I mean, I'm sure you're thinking about ketogenic and maybe broad macro ratios, but how granular?
Yeah, it's a great point. And it's something that we can get a lot more granular than we are right now. Actually had a really interesting conversation with Chris Palmer, who you might know, who wrote Brain Energy, about kind of getting his thoughts on some of these things. So like a good example of that is, so phosphatidylcholine, we think, is a really important molecule. A lot of people do. The digital twin model predicts that a lot of people, this becomes rate limiting. You literally run out of it. Like the simulations run out of this, and then you start having neuronal death. And so we looked in the nutrition literature and just said, do people who eat diets rich in phosphatidylcholine get Alzheimer's later than people who don't? The answer is yeah, about 3 years later on average in these observational nutrition studies. And I will say it's very different to machine learn the nutrition literature than it is to say, here's one focused hypothesis. Does it hold up when you look in? That's a very different thing. So that I think is very interesting. So then we started to look at, so right now, the way that we're doing it, it's very granular. It's not, it's pretty high level, which is that we ask questionnaires about the kinds of foods that people eat. And we basically bucket them into 4 groups of, high, medium, low phosphatidylcholine, and that we use that as a crude measure in the model. Now, we'd much rather, as we get the metabolomics test going, say, all right, let's look at the actual blood measures and get it more that way. And a better one would be a tissue measure and so forth. Anyway, all I want to say is that there's so much granularity that we can get in terms of understanding that person's diet. And some things we're at a very rudimentary level, and some were at a really advanced level. Like glucose, you're at a very advanced level because you can wear a CGM and you can monitor it like crazy. And so there's this continuum like that. So that's an area that I'm really interested in. We're working on, deep metabolomics testing, where we could measure maybe tens of thousands of these small molecules. And my hope is that we're going to be able to learn, partly by tying it to the standard labs, right, as our Rosetta Stone again, but figuring out how do we read tons of different measures off of that kind of, big omics test that you could do inexpensively at home at scale. But as we start to learn how to read that information, we should be able to start informing these things at a much, much deeper level. So it all goes kind of hand in hand. And then the other element that I think is so interesting is that with the technologies like the GPTs, the large language models, we can start to give back, especially if we're doing this with physicians, where there can be two-way dialogue, and the algorithms can actually understand that dialogue. And so we can have conversations at a scale that we kind of couldn't do before. Anyway, that's getting a little bit out there, but it is a really interesting space to tie together multiomics, digital twins that tie that multiomics to patients, and then the AIs that really let us deliver content and information in ways that are accessible and understandable. And I think that's going to really change the future of medicine.
Wow. Yeah, I think so. So the omics data that or the metabolomics data that you're looking at, going back to putting this to nutrition, are, the molecules that are being absorbed and transformed, et cetera, et cetera. So you'll be looking at the influence you'll be looking at the effect of the dietary pattern more than the dietary pattern itself, which is cool. That's
what I would like to get to, because, and there's some work to do, obviously, to get there, but we all know the difficulties of, the questionnaires. No one wants to log their food for long. A few people, but most people won't log their food for long periods of time. There's too much lying and exceptions. And I'll just use myself as an example, right? I've logged my food for periods, but the day I'm most likely to not log is the day that I fall off the wagon, right? You're like, I'll start logging again tomorrow. Today was bad. I was stressed or whatever. And so that's a very human response. And so and there's some clear examples, like in the Pioneer 100, one of the interesting molecules is codenine. Codenine tells you with basically 100% accuracy if you smoke, because you look for codenine in the blood and it's 0, 0, 0, 1,000, 0, 0, 0, 1,000. Because if you're smoking, it's in there. If you're not smoking, a few people would, maybe if you're chronically exposed to secondhand smoke or something, you'd get, some of it in there. But it's a very clean readout. Now, foods are more complicated, but we should be able to tell the difference pretty well between, are you eating a lot of foods that have polyphenols? Well, then polyphenols will be in your blood.
Huge. Yeah, exactly. Right.
The biggest confounder is that like your recent diet probably matters more than the long term, and we've got to figure out. But there's also, as you know, food frequency questionnaires. The 1-day questionnaire is actually validated pretty well, because if you take any person's like 1 day, it's about 80% accurate, I think is the number, in terms of, because you tend to eat similar at the same time.
It's fascinating. I want to ask you about the factoring the microbiome in there as well, but I want to let me comment on the microbiome. Go ahead.
Comment on the microbiome
I'm thinking about the transformation of our nutrients, macronutrients through the microbiome, and then the polyphenols and the postbiotic compounds that are absorbed and all of that. So, in theory, if your food frequency questionnaire is BS, you're going to just obviously have a much different picture. So I'm just wondering, but I guess maybe more fundamentally, my question is, factoring the microbiome influence on everything into the twin, into the digital twin.
Yeah, we haven't done that in yet, but it's going to be really important. Like the one area that we have looked at, and it's a very important one. So phosphatidylcholine I mentioned, right? So we think that's really important for brain health. Model bears that out. The nutrition data bears that out.
I've got some right here, but yeah, go ahead.
And there is a bot here. If you have certain bacteria, and there's like 30 different bacteria, I think, that are known for now, they will actually eat phosphatidylcholine, and they'll turn it into trimethylamine. Now, you'll probably know this, right? Because trimethylamine gets in your blood, your liver turns it into TMAO. TMAO is a risk factor for cardiovascular disease. So you could be pounding a supplement or eating lots of eggs. You're trying to get your phosphatidylcholine in the brain, which is a very smart, good thing to do. But if you actually have the wrong species of bacteria, you're getting a different compound, and that's not good.
Mm-hmm.
So some of the ways you can monitor for that. So, I definitely recommend monitoring, TMAO periodically, especially if you're taking phosphatidylcholine or something like that. I'm going to get mine measured again in a couple of weeks. Also, just to look for that. And then you can see if it's in your microbiome, ways that you might then, try to shift that away.
Mm-hmm.
And so one of the things that I'd love to get into the twins and into some of the things we do with clinicians is to have, like a streamlined approach that's monitoring for that, along with ways that you might, that you could really go about trying to remedy that. So that gets into an integrated approach. And it is important to think about, because the microbiome, every single thing you take into your body, right, whether it's a supplement, food, or a drug, can get modified by the microbiome. And there was a great Cell paper a couple of years ago. So about 13% of drugs are metabolized away by the microbiome. And it's a different 13% for different people. It depends on your microbiome. And they did this really deep dive. So that means you could be on a clinical trial, or just, on a drug from your physician, and you're on something totally different. And you don't know that because the microbiome is 2 black box. So that's why when we say the microbiome is so important for health, it really is. And the depth that we have now compared to, when we started Aravale like 9 years ago, radically better. And microbiome in 5 years are going to be radically better than they are now. So we're learning, but a lot of this has become so actionable now. Mm-hmm. And I really think anyone that's not paying attention to microbiome is doing their patients or a disservice at this point, because it's a critical factor to so many things.
Let me just ask you this. Let me go back to TMAO. So, Bob Browntree always talks about it as being, surrogate marker of choline insufficiency. So if you've got a bunch of TMAO, your choline in circulation is probably low.
That's probably true.
because it's being commandeered by the microbiome. You could actually look at that with your data set. You could go dive right into that and answer Bob's question. We talk about, he brings this up all the time. We were just talking about this a little while ago, but anyway.
Well, just one quick aside, that is the beauty of these really dense data sets, the kind we built at Airville. It's exactly this.
So
you have
answer
this
the question.
And usually you say, oh, okay, I got to go raise money. I got to go do a clinical trial. I got to go measure. So as long as you're doing these smaller data trials, but when you have something like what we had in the Airville data set, you can literally know that by tomorrow.
Go query it, yeah.
Go query it. That's why it was the most fun project that I ever did by a million miles, because you could just ask the question. Well, I'll give one aside, and then I want to come back to your question. So the big effect on statins, for example. So what, and this is work again with Sean Gibbons at ISB, but we took the Airville data because tons of people are on statins and we have their microbiomes, we have all this information. And we looked, and we take microbiomes, we just cluster them into 4 groups, just data driven, and then analyze statin results for those different groups. And it turns out that if you have a microbiome in this group that's high in Bacteroides, those people get double the lowering of LDL cholesterol on the same dosage of statin.
Amazing.
Double the effect.
Wow.
Now, the other side is that the downside of statins, or one of them, is that you get nearly 10% increase in the rate of diabetes. Well, our study also has all the diabetes markers, right? We have hemoglobin A1C and fasted glucose.
Yeah.
So we looked at the same influence of the microbiome, and 2 of the 4 groups, you get a significant jump up in the diabetes markers. But in 2 of them, they're not statistically different at all. So you can segment by the microbiome people who are at risk for potentially transitioning to diabetes from those that aren't.
Statin-related diabetes. Wow.
Who is probably going to be the most efficacious. Unfortunately, there is an overlap between efficacious and having the diabetes side effects. So there's not like one area that's overwhelmingly, the answer.
But you could go into it with your eyes open.
You can, and you can understand it. And that's just finding it, right? Now, we might be able, by diving in, to really understand what those transformations are and optimize it for somebody.
Yeah. So what do we do now? Like, how do we answer that question? How do we know? What do we look at in the microbiome to determine? It's interesting. I have a patient whose calcium score just came back and it's over 1500. So she's at really high risk. I mean, she's going to have to be medically managed for that, and in part, and maybe other things, but like, how do I discern whether she's going to be a favorable responder or she's going to develop diabetes? I mean, how do I actually obtain that information from a stool test?
Yeah, I mean, I think the best way, yeah, you can do it. Yeah, we're looking at, we've got to get that on to the market to make it easy. If you have as I assume most doctors have, an in-house microbiome computational person that can,
crunch some numbers for us. Yeah.
Rhonda. Yeah, exactly. It's totally easy. So this is one of the reasons when I moved to Thorne, I'm like, I got to get more of these things like out in the market to make it easy for people to do this stuff. So we can develop, that. ISB did patent that, so they are trying to license this out to get a commercial partner to build this into a tool for doctors. So you can do that. Of course, the way to do it right now is to monitor for those diabetes markers. I think anyone on a statin right now watch for that. Yeah. And then in the microbiome, you could look for, you can look for this high abundance of Bacteroides, like some of the features that are in, but that's not so hard to see. Right. And so you could look for that. And if it looks like they're in that clade, then that might be a reason to say, okay, well, the microbiome might be partially driving this. Mm-hmm. And are there steps then and then you could look into, diets, certain fibers and so forth to try to shift to a different microbiome and then update. I think there would be a fair amount of trial and error at this point in terms, but you can try interventions that are very,
it's a creative way to look at a stool test that, that most of us in functional medicine are getting. All right. So I'm going to just circle back to TMAO again. Like, is this a surrogate? So I talked about it being, or Bob is the one who suggested it as a surrogate marker of choline deficiency. So it's being hogged by the microbiome. Is it a surrogate marker of dysbiosis also? I mean, are the players who are banging out the TMA, are they dysbiotic players?
They are. They are certainly. I don't know if I would go so far as to say I mean, they are dysbiotic in the sense that they're making TMAO.
TMAO, right.
And so I'm not exactly sure, how far I would push that in any of their particular cases. They are definitely dysbiotic in the sense that you want them gone. And there's no real, as far as I know, there's no positive benefit to them. So, it's not like when you're getting rid of those bacteria, as far as I understand the science today, you can get rid of them. A score of 0 is the best score to have on them. So it's not like they're doing all these positive things and then there's a side effect of TMAO. As far as I know, those things can all be filled in by other bacteria that won't make that substance.
That's really interesting. Okay. And your stool test actually tracks all those, the TMAO players? Are you?
The Thorne test does not do that in as much detail as I would like it to yet.
Okay. And can you just name a couple that I might be familiar with? The listeners might be familiar with.
We had
No pressure.
We had a great version of it at Airvale. I'm not sure who else's test would do better on that, honestly. So I'm not aware of one that does it really well. We built that at Airvale when I was there. It's the one. It was like one of the very first things we did.
Wow.
We will have it on the next version at Thorne because I moved here and I want it on there. And so, so that's coming out.
We'll stay tuned.
I'll go for that. Yeah.
Yeah. I mean, in the meantime, we'll measure TMAO. So there was a paper that came out recently that you were associated with. I know you were an author on it, but you were associated with it. And I know it's born out of work that you've been involved with forever. And it's a remotely coached multimodal lifestyle intervention for Alzheimer's disease, ameliorates functional and cognitive outcomes. That this just came out. So you're looking carefully at, I'm assuming that they were using the digital, I'm wondering if they were using the digital twin model, but I want to just anchor that back to your book where there's a pretty moving chapter that Lee Hood wrote about his wife and her descent into Alzheimer's. It's really moving. And then, obviously you guys go into, extensively your thoughts on what we need to do to preserve cognition as we're aging. But anyway, so tie that all together. This paper, is this using the digital twin, and just appreciate the chapters in the book.
Yeah, so the paper is not using the digital twin, and that's partly it's just the timing of these things. So I had a pretty big role in initiating and launching the work that led to that trial. But then when I moved from ISB to Thorne, then I was less involved in the details of that trial. So I think I'm listed as part of one of the consortium members or something like that. But, so Jared Roach really led out that trial, and that trial tried to keep things very simple. So it was, and it became remote during COVID. And at that point I had moved to New York, I think, during, this period. So what they did was, it was remote health coaching, dietary and lifestyle advice, and brain training, cognitive exercises, basically. And so those things were used, and what they were able to show was very much like, in the spirit of Finger and, and the more recent, like the Four Winds trial and things like this, basically shows that you get more benefit from these kind of lifestyle interventions than any known pharmacological agent. And it showed an ability to, but it was still just slow the rate of decline in those cases. It was almost flat, the group that was treated, but it was slightly down. And then there was the untreated group that came down more. So again, just showing, that dementia is a lifestyle modifiable. The digital twin stuff was, and what's in that chapter of the book, so there's Lee's story. Then the next chapter was really mostly about the kind of work that we did on the digital twin models after I moved to Thorne. That's the area where we really got into that. So it would be great if that trial were like the trial of the digital twins, but we were actually building the digital twin model contemporaneous with that trial. In fact, we started the trial earlier because the trial initially was going to be a straight up test of the Airvale program on dementia. And so it was kept to the same set of measurements that we were using in Airvale and the coaches. Jennifer Lovejoy was very involved in that back then as well. And so that was the way that was set up. And so I think it came out as, another example that multimodal works even if done remotely. The health coaching just makes a difference. And it gets into one of my real pet peeve topics, which is, that for so long, especially when genetics first started coming out, people would repeat over and over again, don't go find out about your genetics because you'll learn you have high risk for Alzheimer's and there's nothing you can do. And it's such a lie because, what was always meant by that is there is no drug that's approved that has any sort of useful treatment for Alzheimer's, which is true. But for prevention, you can do so much. And for people, for listeners that are interested, I did write this piece for the LA Times a few months ago on arguing that the return on investment or the focus on treatment over prevention in Alzheimer's has been really a disaster because we've spent hundreds of billions of dollars at least on especially anti-amyloid therapies and got almost nothing in return. And yet prevention is in a totally different universe. And I do want people just to think about this. Maybe it's obvious if you do. Once you've lost your neurons and synapses, the notion that a small molecule is going to regrow them, bring them back, totally fanciful as far as I can tell. I don't know what mechanism that would possibly do that. But prevention, keeping those neurons from dying in the first place, way easier, much more doable, very possible problem. And of course, we've all known this since we were 3 years old. This is the story of Humpty Dumpty. So it's not, once it's broken, hard to fix. Keeping it from breaking is much, much more reasonable. And so I think that's the thing, so coming back to that genetics, find out, in other words, it's not don't find out your Alzheimer's because there's nothing you can do. Find out because there's so much you can do on prevention. And some of these are so easy: vitamin D, phosphatidylcholine, omega-3s, DHA, phosphatidylserine, exercise, oxygenation to your brain is massively huge. Get out, run, jump around, lift weights. All this stuff makes a huge, huge difference to your brain health over time. And so that's one of the things I think is such an important message just to get out there is Alzheimer's is not, it's not fatalistic. There's not an infinite amount of room to push it off, especially if you have the bad genetics. But honestly, if you push it off, there's other things. I was listening to these lectures on genome writing from Flagship Pioneering recently. An old colleague from grad school days, Jeffrey von Maltzen, was talking about this and some other people. But, as that gets better, in our lifetimes, we're probably going to be able to rewrite your ApoE4. We can probably get rid of it. We have to see. So you want to be doing all these things so you extend your time period because, then your envelope could change radically. You could get an ApoE2 and all of a sudden Alzheimer's is not going to happen until you're 100 or more. And we all know that there's that possibility as we get onto these exponentially rising improvements.
Boy, we are at an extraordinary time in science and medicine. God, so glad we had this conversation. Thank you. I have a ton more questions. I will just have to circle back. Thank you so much for your time today.
Yeah, thank you, Cara. Really, really great to be with you. I really appreciate it.
As always, thank you for listening to New Frontiers in Functional Medicine, where our sponsors help bring the very best minds in functional medicine. And of course, today is no exception. Not everybody can be a sponsor on my platform. I appreciate the good work, the relentless research, and the generous support from my friends at Rupa Health, Biotics, Integrative Therapeutics, Timeline, Mira, and OneSkin. These are brands I know and trust and use in my clinic and can confidently recommend to you. Please visit them online and let them know you learned about them on New Frontiers. And if it's not too much to ask, consider leaving a thumbs up and a kind review wherever you're listening to New Frontiers. Thanks.